Hi everyone. In this explainer, we're going to be looking at a novel technique that gives us a high-resolution view of the interplay between DNA and RNA inside a single cell. Our focus will be on how this is being applied to better understand the aging human brain. You see, as our cells get older, the way their DNA is packaged, this intricate 3D structure we call chromatin, it undergoes some pretty significant changes. And what's interesting is that these changes can vary quite a bit from one cell to the next, even within the very same tissue.
So, this leads us to a central question in cell biology. How does that 3D genome structure actually relate to which genes are turned on or off in a single aging cell? Historically, our methods for analyzing tissues involved grinding everything up, which meant we were just looking at the average of all the cells. This process completely masked the critical differences between individual cells. So, to really get at the relationship between genome structure and function, we need to be able to profile both of them at the same time within a single cell.
All right, here's the roadmap for our discussion. We'll start by outlining the existing technological gap in single-cell genomics. Then we'll introduce the new technique designed to fill that gap. After that, we'll look at how it was validated, and finally, we'll dive into the key biological findings that came out of its application to the aging human cortex. Even with some of our powerful single-cell technologies, we've been missing part of the picture. While some methods can look at chromatin shape and gene expression together, they really struggle to simultaneously profile multiplex chromatin interactions, gene expression, and RNA-chromatin associations all within one nucleus. This gap has limited just how well we can understand the link between the genome's structure and its function at that single-cell level. And understanding these multiplex interactions is really critical. You see, the genome's architecture isn't just about simple one-to-one contacts between two bits of DNA. It's often much more complex, with these higher-order structures where multiple DNA sequences come together in 3D space. Mapping these multi-party interactions is essential if we want an accurate model of how the genome is organized. So, to address this technological need, researchers developed a method called MUSIC, which stands for multinucleic acid interaction mapping in single cells. It's specifically designed to concurrently profile all three of those modalities: multiplex chromatin interactions, gene expression, and RNA-chromatin associations to give us a truly integrated view of what's happening inside the nucleus. Okay, so how does MUSIC actually work? Well, it relies on a very clever, multi-layered barcoding strategy. First, to tell RNA and DNA apart, they add distinct linker sequences to each. Second, to make sure they can trace every molecule back to its original cell, they add a unique cell barcode using a process called split pooling. And third, to identify those multipartite contacts, each molecular complex gets its own unique barcode. This whole system ensures that every sequence and interaction can be accurately pinpointed to its specific cell and molecular complex of origin.
Now, before you apply a new technique to something as complex as the human brain, you have to be absolutely certain it's accurate. So the method was rigorously validated using a mix of human and mouse embryonic stem cells. By comparing the data from MUSIC to establish methods that only measure one thing at a time, they could confirm its precision. This mixed species experiment was a key control, and it showed very low rates of mixing between human and mouse molecules, which supports MUSIC's ability to generate reliable data.
And the results of these validation experiments were quite strong. The DNA contact maps that MUSIC generated were a good match for those from microC, which is the gold standard method, reproducing the same key structures. On top of that, the gene expression levels measured by MUSIC showed a high Spearman correlation with both bulk RNA sequencing and another method called iMARGI. This concordance confirmed that MUSIC is providing reliable data for both chromatin structure and gene expression. A key observation from the validation was just how much better the multiplex data was. When the researchers looked only at simple pairwise DNA contacts, they couldn't clearly see well-known structures called topologically associating domains, or TADs. These are like functional neighborhoods in the genome. However, when they used the multiplex interactions, the ones involving 3 or more DNA reads, the TAD structure came through loud and clear. This really underscores how important it is to map these higher-order structures.
All right, with the method validated, it was time to apply it to the aging human brain. The team generated a MUSIC dataset from 14 post-mortem frontal cortex samples, all from tissue donors who were 59 years of age or older. In total, this dataset gave them 9,087 single nuclei to analyze.
The RNA component of the MUSIC data immediately allowed for a robust classification of all these nuclei into their distinct cell types. Based on the single-nucleus RNA profiles, they were able to identify 7 major types, including excitatory and inhibitory neurons and glial cells like astrocytes and oligodendrocytes. This detailed cellular map then became the foundation for all the cell-type specific analyses that followed. The analysis then shifted to look at the variation in chromatin structure across all these individual cells, and how that might relate to aging and disease. What the data revealed was a high degree of variability in chromatin organization even within a single cell type, and this variability seems to have real functional consequences. To quantify this variability, a new metric was developed, the Local Chromatin Structure Erosion Score, or LCS erosion score for short. This score essentially boils down all the complex chromatin contact data from a single cell into one value. It's derived from the curve that plots how often different parts of the chromatin interact based on their distance. A higher LCS erosion score basically means a greater loss of that local, short-range chromatin structure. Cells with high scores are termed LCS eroded. And this is where a significant finding emerged. When this new chromatin metric was compared to the cell's molecular state, they saw a clear pattern. Using a published model, they calculated a transcriptomic age for each cell from its gene expression data. And consistently, across every single cell type, the cells they classified as LCS eroded had a significantly higher transcriptomic age. This suggests a direct link between the breakdown of local chromatin structure and the molecular signs of aging, leading to this new concept of a chromatin conformational age. This connection was then backed up by a direct correlation with the disease state of the tissue donors. It turned out that 7 of the 14 donors had high Alzheimer's disease pathology, as defined by a break score of 4 or higher. The analysis showed that single cells from these high pathology samples had significantly higher LCS erosion scores than cells from low pathology samples. This suggests that this disorganization of local chromatin is a prominent feature of the Alzheimer's disease state.
The data also uncovered a significant sex difference in how these cells were aging. Based on that chromatin conformational age, cells from female donors showed a different aging pattern than those from male donors. Specifically, the female cortex had fewer old neurons, but a much higher proportion of old oligodendrocytes. In fact, the odds of seeing this particular imbalance were almost 7 times higher in females. This might have implications for understanding sex-specific susceptibility to certain neurodegenerative disorders.
So, to investigate these sex differences more deeply, the researchers leveraged music's ability to map RNA-chromatin associations. The analysis zeroed in on the interactions of the XISIT long non-coding RNA in female cells. XISIT is known to be the master regulator of X chromosome inactivation. The music data showed 2 completely distinct conformational states for the X chromosome in female cells. Chromatin that was associated with the XISD RNA, which corresponds to the inactive X chromosome, had fewer short-range contacts and more long-range ones, consistent with a very compact structure. On the other hand, chromatin that was not associated with XIST, the active X chromosome, showed more frequent short-range contacts and had a structure much more like the other chromosomes. The analysis showed significant cell-to-cell variation in how much exist was associated with the X chromosome, a metric they termed XAL. Importantly, the structural difference between the active and inactive X chromosomes grew larger with higher XAL. And this effect wasn't uniform across all cell types. The structural difference was most pronounced in excitatory neurons. This points to a cell-type-specific mechanism for regulating the X chromosome, which could be a molecular basis for some of the known sex differences in brain function and disease. So, to summarize, the music technology provides an integrated, multi-layered look into single nuclei. This has led to the concept of a chromatin conformational age, which is a potential cellular biomarker linked to transcriptomic aging and Alzheimer's pathology. The method also uncovered cell-type-specific mechanisms of X chromosome regulation, which offers a potential molecular basis for sex-specific differences in brain health.
This work leaves us with a compelling question. Could the chromatin conformational age of an individual cell actually be used to predict its future health? Could this physical state of the genome tell us something about a cell's functional trajectory or its vulnerability to disease? This new metric gives us a powerful new access for investigating the fundamental processes of aging and neurodegeneration, all at the ultimate resolution of a single cell.